Thank you, Ryan. Okay, good morning, everyone. Thank you for the introduction, and I'm very happy to be here. Very glad that you guys were able to attend today. I'm gonna give an update on soybean disease management. I feel like I have been talking about these diseases quite a lot this winter, and I think it's a good time to do it. So again, my name is Horacio Lopez-Nicora, assistant professor in the Department of Plant Pathology. I am the soybean pathologist and nematologist, and this is the group of diseases that I want to present on. Soybeans is nematode, sudden death syndrome (SDS), just an update, and then red crown rot, which is an emerging disease. And we'll see if we have time for maybe a bonus on an extra soybean pathology project that we're working on. Just a reminder, this is like the basic plant pathology 101 disease triangle. For us to have a disease in the field, we absolutely need three elements. The virulent pathogen, it has to be virulent, right? It has to be virulent. A susceptible host, it has to be susceptible. In this case, we're going to talk about soybean because that's what we care the most. And then the third one is a conducive environmental condition. And sometimes that's one that we can alter and modify and change if we have these two other pieces already in our field. So we can have a virulent pathogen in our field, we can have a host in our field, but if that conducive conditions are not present, we don't have disease. And that's sometimes important when we are thinking about management. For SEN, I'm gonna just walk through the life cycle very quickly to understand some important part of the biology of this animal. It is an animal. It overwinters. It stays alive in the field as eggs, and that's what we want to quantify when we send a soil sample to the lab. Once your field get infested with SCN and pretty much with any other soil-borne pathogen, you cannot eradicate them anymore. You cannot get rid of them anymore. You will have to manage them. So the nematode will hatch from the egg into this second stage juvenile. Is the only virulent stage in the life of the nematode, the only stage in the life of the nematode that will infect the roots, migrate to the vascular cylinder. So this is a secondary root. You can see that all the little thread-like nematodes are going inside and then migrating to the vascular cylinder. We stain these roots, right? We stain with a dye that can stain animal tissue, and then we can bleach and clear the root so we can track what the nematode is doing. The nematode will start feeding sites in the root and become sedentary. It will not move anymore. Those cells around the head of the nematode will feed the nematode for the rest of its life. The males will regain this vermiform, worm-like condition and leave the root to fertilize the female. They won't feed anymore. They will fertilize the female, and then the female, on average, will lay 250 eggs. And this can happen. This can happen 3 to 6 times in a season. So very rapidly, we can jump from very low levels of SEN to above damage threshold. The roots won't lie. If we pull those roots from infested fields, they will show the female attached. So these roots got infected with the nematode, and then the female protrude from the roots, but they stay attached. Remember, this is a sedentary pathogen, cannot move. And then you can see the nodule here, just showing the nodule, those nitrogen-fixing symbiosis with bacteria, very large, very big compared to the soybean cyst nematode. We want to see a lot of nodules. We don't want to see a lot of soybean cyst nematode. In situations where we have stressors in the field, other abiotic stresses, such as, for example, nutrient deficiency, potassium deficiency, to put an example, and soybean cyst nematode, we may see more severe damage of that or symptoms of that nutrient deficiency. So marginal chlorosis will be more evident. We will take a soil sample, send it to a soil lab. Result will come that we need potassium. We will add it, and then the next year we will not see any of those symptoms anymore. We fixed nutrient deficiency. However, if we fail to send a subsample to the nematology lab, and the populations of the nematodes will continue increasing in that field. And yield reduction, as a consequence, will continue happening. Sandy soils, some people say, well, sandy soils are more prone to severe damage of nematodes. Yes, but it is because we are adding additional stressors, evapotranspiration and things like that, that will, with infection of the nematode, reduce the growth of the plant. So we can see here a resistant versus a susceptible, we can see standingness, etc. Presence of other pathogens can cause interaction. SDS, for example, is a well-studied um, disease complex, right? When you see a lot of SDS symptoms in the field, maybe that field is infested with SCN as well. But, however, having said all these and how we can possibly recognize that we have SCN in our field, this is the most typical symptoms that we will see in the Midwest, in high soybean producing regions. Absolutely no visible symptoms. The field look very healthy. Half of this field is susceptible to SCN, the other half is resistant. We don't see any visible symptoms, yet when we go and harvest this half, we can see significant yield reduction due to the nematode. And I'm talking about above 30% yield reduction when fields look very healthy. So, you have white mold, and white mold will tell you is there. White, fluffy mycelia, the plants are dying. They're probably smelling already when you walk that field. SDS will tell you it's there. It's going to show you that intervenial chlorosis is very evident. You name it. All these other diseases cause symptomology that we can associate and use to diagnose the problem. With SCN, we don't have that in many cases. The field looks really good. I'm very happy with how my field look, and we're living below ground over 30% of the yield. That's a lot. So how do we manage something that we cannot see? Well, a soil test is the best way, the best and the proactive way of managing SCN. Test your field, know if you have it, know how much you have it, and we actually help with that. With support from Ohio Soybean Council, we process up to 2 samples free of charge for Ohio growers. You can submit this to the lab, and we will process those samples to you and report the result in numbers of eggs per 100 cc of soil. And I will put a QR code at the end of the presentation so you can access to the forms that you need when you collect the samples and send it to the lab. You could also pull the roots and check for soybean cyst nematode. I normally don't recommend this if this is the first time that you're trying to know if you have SCN. I normally use this technique when I know a site that has a lot of SCN and I want to see if one of the treatments I'm using are having some type of effect in reducing the reproduction of the nematode. And the reason is because you can pick a plant loaded with SCN and just a couple of feet away from that, they're not there. So they're very aggregated in the soil, in the field. Okay. Soil test, the best way, proactive way of knowing if you have soybean cyst nematode. And we know how to collect the soil sample. We do it already to send it to the soil lab. It's the same way. You just get that sample that you're going to send to your soil lab and mix it very well and pull a subsample that you can send to the nematology lab. As easy as that. And you can do it at the same time. You probably do it every 3-4 years. That will be a great time to check for your nematodes. Normally, I recommend growers to target areas that could be more prone to have higher numbers, entryways. Anything that moves soils will move SCN. But even like those sites that consistently have been yielding low, why? I have done everything and still that site is consistently yielding very low. Maybe it's SCN. So we run the samples you send to us, and then we say that I report that number to you. We send back, X amount of X per 100 cc of soil, cubic centimeters of soil. Very standard volume, that you can compare across different labs. But what does that mean? I mean, we provide a table in Ohio from our data and evidence. We provide a table that can place that number. in either low, moderate, or high. But when we started to look at different tables from across United States, from other institutions, from other labs, things are not in agreement. What in Ohio we call it low, in a different lab we'll call it moderate. What we call it high, maybe in Iowa they will call it moderate. So the profit checker that was launched last year by the SCN Coalition, which I'm a co-lead of, um, uses over 35,000 research plot data to place your SCN and other parameters from your field and actually have a more data-driven estimate of yield reduction in your field. So let's say you get back your results and you have 1,500 X per 100 cc of soil from your field. You will add that. You will also add percentage of sand that you will get from your physical texture analysis of your soil. You will add your soil pH. You get that from your soil analysis. And then there's a component here that is called the female index on PI 88788. Well, PI 88788 is the most common source of resistance to SCN. Most of our commercially available soybean have this source of resistance, PI 88788. Female index means the ability of that population to reproduce on PI 88788 at a percentage compared to a susceptible. So what does that mean? 33 in this case. So that means that SCN population can reproduce 33% on 88788, that source of resistance, compared to a susceptible. Well, in Ohio, we have growers that unfortunately have SCN populations that can reproduce at 85% on 88788 on this source of resistance compared to a susceptible. It's not resistant anymore, right? Well, that value you do need to send it to a lab that can do that. Our lab can do that. It charges for that service, and it takes a little bit of time, longer than just getting the number here. But it's very valuable to refine what this calculator will give you. If you don't have that value, don't worry. We have provided an average from Ohio. So you can go and you hit Ohio and it will show there and it will populate the female index on 88788 from Ohio. If you're farming somewhere else or working with a grower in a different state, you just select that state and it will auto-populate it as well. But at the end, the calculator spits a potential yield loss due to that population under those conditions, and also an economic loss. So a pretty good tool that I recommend you using it when you're trying to discuss results with your growers or yourself, right? If you're a grower. And because we receive these samples to the lab and we're able to trace it back, and you can see since I joined in 2021, we have been processing over 400 samples every year. We can update our SCN distribution map in Ohio. And not only distribution, but also how much on average we have per county. And this, we're very grateful for the support of Ohio Soybean Council, of course. It also allows us to track different counties that before we did not detect SCN, and now for the first time, we are detecting and we know we have it. Over the 2,200 samples that we have processed so far, more than 60% are positive for SCN. So it's not just flipping a coin here anymore. It's whoever comes and join this meeting today possibly have SCN with more than 60% certainty, right? So it's not just flipping a coin anymore. We have SCN in Ohio, and pretty likely that we have on the next field that we go and sample. Fortunately, though, most of those that tested positive for SCN have low levels of SCN. And keeping those low levels low is the best strategy than trying to bring very high levels below damage threshold. Now, the question that you probably are asking yourself is, well, are all these populations behaving the same? Because I just say, well, I work with a grower that have a population that can reproduce by way more on 88-1088, but can they reproduce on other type of resistance? Well, we have to go back a little bit and understand what does that mean. When we discovered soybeans as nematode in the United States, 1954, we soon after started identifying sources of resistance. And these are the different lines. PI stands for plant introductions. These are horrible seeds to grow. These are soybeans that are black coated seed, and they grow very viney. You will never grow it for commercial purpose, but they have traits that we can introgress in commercial soybeans, right? And then the number here, 1 through 7, is a chronological order when they were discovered. So Peking was the first one to be discovered, did not allow the nematode to reproduce. 88-1088, the most common source of resistance, was then discovered, and so on until cloud. Now, 88-1088 was very easy to introgress in high-yielding soybeans. Soybeans we liked to grow, soybeans we knew how to grow, soybeans we really like how it yield as well, right? And it did the work. It really did the work on that population of nematode. We have AG type 0 and 0 because it cannot reproduce on any of those 7 sources of resistance. So notice how PI 88-1088 was actually avoiding the reproduction of the nematode compared to a susceptible. The nematode, that population, was not able to reproduce above 10% compared to a susceptible. And that's what makes that population type a type 0. It did not reproduce on 88-1088 or on Peking number 1 or on cloud number 7 or on any of the sources of resistance. AG type 0. Now, it works so well that we put it and we introgress this source of resistance in pretty much 90% of the commercially available soybean. So everyone was planting 88-1088 for over 30 years, nonstop, on the same population. And that exerted a selection pressure that shifted the virulence of an AG type 0 to an AG type 2 that now can reproduce above 10% compared to a susceptible. And like I just mentioned, we have populations in Ohio that can reproduce 85% on PI 88-1088 compared to a susceptible. And we call it type 2 because it can reproduce above 10% to a susceptible on that number 2. So a population that can reproduce on Peking, we're going to call it type 1. Okay. So Gili Correa, a PhD student in my lab, found data from 30 years ago in Ohio. They phenotyped the populations, and now he phenotyped 150 populations in Ohio. So we wanted to know what type they were, type 0, 1, 2. And you can see that 30 years ago, the map was more blue. You can see that there's less of that light blue in our current map of Ohio. And if I just show it with a graph, it make a little bit more sense, right? That bar is going down where these bars are going up because of the recurrent use of the same source of resistance. And you can see that now the predominant population in Ohio is this type 2, which by definition can reproduce on PI 88-1088. And this one here, the green one, is my nightmare. My nightmare because it's a population of soybeans with nematode that can reproduce on 1 and 2, which means that it can reproduce on Peking above 10% compared to a susceptible and on PI 88-1088. And those are the 2 commercially available sources of resistance for SCN. So we're in trouble. Okay, management for SCN, integrated management. Once you have it, you cannot get rid of it. Know if you have it, sample your field again to monitor those numbers that they're going down or they're staying low. Rotate sources of resistance. Don't feed the same genetic material to this animal. Rotate sources of resistance. If you plant PI 88-1088, plant a Peking on the next time you do soybean. If you cannot find a different source of resistance, switch varieties. Don't train this animal to eat on the same genetic material. Rotate to a non-host, corn, wheat, and consider a seed protectant because this will enhance the root health overall. And then this is a 6 years rotation that we proposed. PI 88788, very easy to find. Go to a non-host corn. Go and find a Peking, something different. Rotate sources of resistance here. Go to corn again. And then try to find something different than Peking. Never plant Peking twice in a row. It took us 30 years to adapt our population to PI 88788. We have evidence that it will take us way, way less than that to have a population adapt on Peking, unfortunately. The resistance on Peking works so well, but if a couple of those nematodes can adapt, then they will overcome that very quickly. And then we go back here in year 6 to corn, and then we repeat the cycle. Okay. We'll transition. We'll stay in the root system still. We're going to talk about soil-borne pathogens still and give a little bit of update on SCN. SDS, sorry, sudden death syndrome. Yeah, visit to a grower. You can see yellow patches here. You can see green and yellow in between. You can see that area here is actually prematurely plants that have been defoliated due to this pathogen. This is at the R5 stage where the plant has to put a lot of weight on those pods. We have no leaves. There's no photosynthesis then being produced. Clearly, you can imagine that this is the reason why we're losing yield. Then the fungus is in the root system, damaging the root system. The fungus actually infected the plant very early at planting. And if we planted in cool soil temperatures below 65 degrees Fahrenheit, the damage is more severe due to SDS. Now, the fungus produces a toxin that then is translocated upwards and presents that symptoms in the leaf that causes defoliation. So there's actually two big stress that are happening to the plant in the root system, where the fungus is rotting the root system, and on the above ground part where the fungus is sending toxin to defoliate the plants, essentially sending a herbicide up there, right? Because we won't find the fungus in the foliage. And this is a typical symptom of SDS, green veins, and in between the tissue is dying or very yellow. If we pull the roots, we can actually see the blue growth of the fungus there. And that's where the damage is taking place. The fungus is in the root system. You send me leaves and ask me if this is a fungus that causes SDS. I cannot pull anything from those leaves. The fungus is in the root system. The toxin is produced in the root system and translocated above ground to cause these symptoms. Now, unfortunately, these type of symptoms can be caused by other pathogens. Brown stem rot is a disease caused by a different pathogen, will have the same type of symptoms. Red crown rot is a disease caused by a different pathogen. We're going to talk about red crown rot, but causes exactly the same symptoms. So just by looking at the leaf and seeing these and saying, oh, I have a little bit of SDS or I have SDS, is not enough and sufficient. You need to go there, pull those plants, open those stems, and see if the pith of the stem is healthy looking. That may be SDS. If it's brown, discolored, that could be brown stem rot. If in the root system you can see some of these blue discoloration, the actual fungal growth, you may be more certain that you have SDS. If you instead see red discolorations and red little dots, balls, spherical structures, that may be red crown rot. But this is SDS. The grower we visit clearly was worried about how damaging the disease was that year, and he was concerned because he used a seed treatment. And how is it that my very effective seed treatment is not working for SDS? And we did a little bit of digging, and we saw that the variety that was planted in that field was highly susceptible. So your first line of defense always is partial resistance, and then you can add some effective seed treatments that can help. But seed treatment by itself is not the silver bullet here, or with any soil-borne pathogen. But we had three questions when we visited that farm. We had three questions with Jenna Moore, a PhD student in my lab that graduated last year. What are the species that we have here? There's more than one species that can cause SDS. Are they developing resistance to fungicides, especially the ones that are very good for SDS? and then how aggressive they are. Are they all behaving the same way when we expose them to soybean? So we had fortunately worked with 50 growers that submitted samples to the lab. So we had 50 independent isolates from across 18 counties in Ohio. A little bit of what we did, we received the sample from growers or working with you guys. You send the sample to the lab. We were able to create that pure isolate. And from there, we asked the question, what are the species we have? We also, in the greenhouse, inoculated plants and see how aggressive they are at rotting the root and causing symptoms. And then we essentially expose them to two active ingredients, PD flumetofen, which is the active ingredient of Saltro. It's a good product against SDS. And then fluopyran, another good product, seed treatment for SDS. So we use those two active ingredients and expose our isolates to different doses. And with that, we calculated an EC50. So EC50 is the dose required to reduce 0.5% of the growth of the fungus. So you can imagine that a larger EC50 means that that particular fungus can withstand and tolerate more active ingredient and continue growing. We don't want that. We want the EC50 to be very small. A very tiny dose actually kills and stops the fungus from growing. So those are the three things we did, again, with support from Ohio Soybean Council. What Jenna did was use a spectrophotometer to do this very quickly. She will put all her isolates in this well with a different dose of the products. In this case, I'm going to show the data from the Saltro active ingredient. And she will put it on a Friday and then come back on a Monday and get all the data. So she actually optimized this protocol. And you can see here on the y-axis, we have all our isolates that, by the way, all of them were Fusarium virguliforme, a very prevalent species that cause SDS in North America. But she also identified one additional species. So 2 fields in Ohio had an additional different species that cause SDS. So that's good. We know that it's not just the most common one, which is virguliforme. But what we can see here is that when we expose them to PD flumetofen, that most of these isolates are highly sensitive, and that's what we want. We want to see low levels of this EC50. They're very low, as you can see here, with some of them showing some reduced sensitivity, but overall really good. And then we also did it with fluopyran, and we can see a wider range here, right? This is a product that has been used longer, has more history. Most of these isolates have been maybe exposed more time to the product. And we see that some of the isolates show high sensitivity, however, some more variability. Again, this is in vitro. We're planning to do then a greenhouse and a field study to evaluate how much of the resistance or sensitivity to these products they actually show. Okay, so we answered those 2 questions. In the greenhouse, we also asked the questions about variability in pathogenicity. Very variable. Some isolates will be very damaging to the roots and produce foliar symptoms. Other will be less so. So know the pathogen you're working with. And with that, I wanna show a little bit of the work that we're doing with SDS with other pathologists across the US. So it's a big group. It's a large group. This is a project funded by NCSRP. You can see different states being part of this, and the idea is to have the same protocols and ask the same questions. And most of the time, these questions come from growers, right? So how well is my seed treatment doing? I want to know how well our Illivo, Saltro, Saltera, you name it. We want to compare those. So each one of us take it and do this in our research station. Of course, we do it with help of our OSU research farm managers that we're very grateful for. So we did this for 2 years. We have over 15 locations each year, randomized complete blood design for treatment, and then we have a non-treated check, a base, which is a standard fungicide and insecticide, and then the actual treatments that we have questions about, right? We want to know how they're working with SDS. We infest our plots. We actually put the fungus when we plant, and then we go and we collect a lot of the data. So I'm going to show you this table, and I know this is pretty overwhelming here, a bunch of numbers and letters. So I'm going to walk you through. I have here on the top the two checks, the non-treated check and the base, right? That's going to be what we're going to compare our treatments to. And then we have Ilivo, Saltro, Saltera, Cyway, and a mix of Saltro and Ilivo. So those are our treatments. Some statistics here showing you just the power of our trials, right? So many trials that we are pretty confident with our results here. And then the variables that we collected. So populations, plants per acre, percentage of the root being rotted by the fungus. So we can estimate that. FDX means foliar disease index. That's the symptoms that you see that I show you at the beginning. So we can rate that. And then yield, of course. At the end, we want more yield when we're adding a treatment on an infested field. So now we can start making sense of what we saw, right? There's a significant and statistical difference with plant population. We're seeing more plants coming up when we're protecting those seeds against SDS, which is also a seedling pathogen, right? It's infecting at the seedling stage. It's causing stand reduction. So we see that when we're treating our seeds, we are seeing a difference when we compare to the check. We want to see less root rot, right? When we protect our seeds, we don't want to see that root system rotted. So we're seeing here a significant difference compared to the base when we are treating our seeds with this effective seed treatment, right? Foliar, we don't want to see the symptomology as well, right? When we walk off fields and we treat our fields, we want to see less. We want to see less symptoms than when we haven't treated with a product that is meant to manage SDS. And then at the end of the day, we want to see a return on investment. So we want to see more yield. And statistically, we're seeing a difference here compared to a non-treated check. Okay. So in summary here, before I jump to red crown rot on my final minutes, confirm the problem. I mean, it can be misleading due to the symptomology. Make sure that when you say I have a little bit of SDS, it's actually SDS. When you say I have a little bit of red crown rot, it's actually red crown rot. We're going to talk about that right now, but make sure you know the problem. Manage SCN first. There is an interaction or report on interaction. So we talk about SCN already. Wherever I see SDS sample, I normally take a shovel or a probe and I collect a sample there from that area. And most of the times, high levels of SCNs are paired with my symptoms of SDS. Choose variety with partial resistance. Like I mentioned, and I gave the example at the beginning, seed treatments are not the silver bullet. You need to pair them with partial resistance. You need to pair them with reduction of stress at planting. We cannot, talk about avoid planting in cold soil temperatures. We won't, right? Spring will come and it's gonna be cool. And Laura and Osler were just giving a presentation here on planting date. The more we wait, the more yield reduction we may experience, right? So we want to get our beans in the ground as soon as possible. So if we know we have SDS, confirm the problem, and we can choose a variety with partial resistance, then make sure to consider a seed treatment that is effective to SDS, and then try to work on avoiding other stressful conditions, such as compaction, for example. Make sure that those soils are well drained. Compaction normally will enhance severity of SDS. Okay, integrated approach, that's the key. With that, I'll put this QR code here for you to take a snap of that. These will take you to a fact sheet that we put together through the Crop Protection Network, where we evaluate and compare different products against not only the SDS fungus, but other pathogens as well, including the oomycetes, the water moles, Pythium, Phytophthora. And then we rate, depending on the active ingredient, we rate as excellent, very good, poor, So it will give you a nice list of something that you can base on, right? Well, I don't have SDS in that part of my field. I'm dealing more with Phytophthora epiphyum. Well, this QR code here will take you to a nice table that you can see what products are rated good or excellent for that particular problem that you may have. So take a look at that. And this is work that we do in collaborations with other soybean pathologists across the US. We meet, we share data, we show results, we compare, and then we agree upon how well they're doing across different regions. Okay, with that, and in my last 10 minutes, maybe, Ryan will keep me in track here. We'll talk about red crown rot, an emerging pathogen in the Midwest right now. What does that mean? Well, it's been around since the 70s in the US. In the southeastern United States, killing peanuts. But recently, however, 2018, it showed up in the Midwest where we plant soybean, and it's causing a lot of damage to soybean. It can cause a lot of damage to legumes in general, okay? But in soybean, 2018, showing up, Illinois, Indiana, Missouri, Kentucky have been doing a lot of research already and working and struggling with this disease. And then for the past 2 years, we have been, just being more vigilant about this. And this QR code will take you to a core newsletter that we put together, and you can check others that we do. But then, unfortunately, 2025, we confirm it in Ohio. So Ohio now is unfortunately part of this group, along with Minnesota and Wisconsin in 2025. So those were the three states that reported red crown rot for the first time in their states. And this is where we found it in Delaware, in the middle of the states. Look at this. Indiana, borderline, all the counties with red crown rot. However, we found it here in the center of Ohio, in the middle of Ohio. I mean, I was here. I was sitting here, just waiting to find it here in this area, in this border with Indiana. However, it showed up here in the middle. Why? And the reason that I think that this is what happened is that we have been maybe misdiagnosing. We may have been saying, "Oh, I have a little bit of SDS in that field. I don't have to worry about it." Or we have been walking fields and we have been trained very well to identify Phytophthora symptoms. And we say, "Oh, I have a little bit of Phytophthora, in that field." And it was red crown rot. Or we know how to identify very well charcoal rot. And it was a dry season and we walk on a field, saw a bunch of plants dying and keeping all the leaf attached to the stem. And we say, "Oh, that's a little bit of charcoal rot there." So this is a problem that causes symptoms that resemble very much different diseases that we know very well how to manage and how to recognize. So SDS, well, it produces exactly the same symptomology. Phytophthora, the same type of symptomology. There are subtle differences. SDS normally will defoliate prematurely. Red crown rot is such a good rot rooter, that all those leaves will just dry out on the plant and stay attached to the plant, as you can see here in this image. This is a field that we visit. You can see pretty severely affected, and you can see that reddish discoloration on the crown, on the base of the plant. You need to go below the canopy to actually make a diagnosis here. Here I'm showing you the fruiting bodies, those dots, spherical, ball-looking fruiting bodies that is very diagnostic for us to say, yeah, we might be dealing with red crown rot here. But again, look at this. If I show you this and I just show you the leaf and I say what is this? You probably will say SDS. I would probably say maybe SDS. I would say maybe SDS. But no, this is red crown rot. And then again, those fruiting bodies that may go away. They may go away when it gets drier, and then you have that red discoloration. And when you send this to the lab, we like to do a thorough investigation of what's going on, right? We like to look at all the parts. We like to pull the actual pathogen from the affected area. This is the actual pathogen that causes red crown rot. But look at all the other organisms that we were able to pull from the same plant. So you need an expert to know that this is the fungus that is causing red crown rot. You need to look under the microscope. You need to know that you're looking at the sexual spore of the fungus that causes red crown rot, the asexual spores, the microsclerotia, which is the way that these fungus will overwinter over time and will move through soil movement. And then with all that, you do a DNA extraction and you confirm you have red crown rot, and only then you go and you tell the grower, unfortunately, my friend, you have red crown rot. So here I just want to show you that when we went and visit there, I know Ryan, I have maybe 5 more minutes.
Yep, you
Let
got
me
about
know,
5
please.
more minutes. Yep.
Yeah, that's what I have here. So I'm good. So when we went there, we went and we actually sample areas that were healthy looking, the green area here, and the symptomatic area, and we did this with a PVC rectangular area of 2 square meter. And when we collected yield, we noticed that we saw, and this is all, we hand threshed and we cut it with our hands and took it to the lab, but over 50% yield reduction. I mean, there's still plants that have the pods and seed, you will get some yield there, but more than 50% yield reduction in those patches. And with movement of soil, these will become homogeneous across the field. We're looking and trying to understand what exactly the fungus is doing. All the agronomic components here. We can see that the root rot is really significantly reducing seeds per plant and seed yield, and very positively related to aborted pods. So really that root rotting of this pathogen, such a good root rotter. I mean, it's impressive. With SDS, you go and you grab a symptomatic plant with SDS and you try to pull it, and you still need to apply some force to get it out of the ground. With red crown rot, it was impressive. It would just come out. And certain literature describes the way that it kills the root as a symptom called the pencil roots, because it just leaves your root looking like a pencil. It just consumes all the secondary roots. It's very wild. So what are we going to do? Accurate diagnosis. There are certain labs, and we are working with these labs and we're working with these techniques that will just get the DNA from the tissue of the plant and run what they call a species-specific primers. And it will light up if it's positive, if it's red crown rot. Unfortunately, these primers can cross-react with other organisms. So now it can say positive red crown rot when actually the fungus is not there. So follow up with your lab. Well, you did the test. Awesome. Can you show me if you were able to retrieve the fungus? Were you able to get it, to isolate it from those symptomatic tissues as well? Do some follow-up to make sure that you have red crown rot. If you have it, rotate to a non-host. These will reduce those survival microsclerotia. Fungicide seed treatment, only those labels, and if you have information that they are indeed effective for red crown rot pathogen. Avoid planting in high temperatures. So this is great. We talked that SDS does more severe damage at cool soil temperature. Red crown rot does better at warmer soil temperature. Manage SCN. There's also a report of interaction. No commercial varieties are currently resistant or with partial resistance to red crown rot. And then no foliar applications. I mean, you're gonna see it. It looks bad. However, the fungus is in the root system producing toxins that show up on the above ground. And integrated management. And with that, I'm going to leave this QR code again. This will give you access to a folder that we prepare with a lot of instructions on how to submit samples to the lab. And we're here to help. We are here to receive and work with your samples in case you want to verify that you have or not red crown rot in your field. So check that QR code, a lot of information there, a lot of the forms and how to submit that. So with that, Ryan, I think I'm on time.
All right. Thank you, Rasha. We do have some questions, some of which I think you maybe answered a little bit because they were asked early on.
Yeah.
The first question that popped up is, what seed treatments do you recommend to minimize stressors that can occur in early planting? And I did post the article that you presented from Crop Protection Network.
Yes.
So, any other thoughts
So
on that question?
no, I think that article, yeah, it came later, that article, but that will have a very thorough answer to that question, because you may have rhizome, or you may have Fusarium graminearum that is causing seedling disease. And we have provided their information for that. You may have Phytophthora and Pythium that we didn't talk today, and that fact sheet will have that information.
Okay. The next question we have is, what are your thoughts on using biologicals as a seed treatment, especially when planting early?
This is a great question. And as a matter of fact, when I show everyone the results for 2 years that we did in collaboration with other soybean pathologists from the US, right? We also included a list of biologicals. You know, we have growers that are like, hey, how is this biological working? And what about this one? We had at least five biological treatments there for SDS. Um, and unfortunately, we did not see any significant difference and effect. And that's why I didn't put it in present today. Um, I guess pretty easy to say there was no effect with several biologicals that we tried, and there are many reports out there published already that you can get access to. Um, Dr. Fabiano Colette, a former PhD student in Laura Lindsay's lab, just recently published a couple of papers just showing the effect of biologicals on agronomic components and yields specifically, right? Um, and again, no significant effect. And this is across multiple states and multiple sites.
All right. Thank
But
you. And
more to
go ahead.
do on that. I agree. More to do on that. We need to figure out what's working best and use that as a recommendation.
The last question that's been posted in the Q&A is, did you see much brown stem rot last year, and what can
Mm,
be done to prevent it or minimize it?
yeah, that is a great question. You know, I haven't. We not last year, but what we did receive sample in the lab. You know, you can easily tell apart because you split the stem and you see the brown discoloration. Again, the first line of defense for brown stem rot, and again, I haven't seen it as prevalently as SDS. Um, so that's one comment I want to make. I haven't seen brown stem rot across Ohio like I see SDS, for example. But if that is the case in your field, then I strongly recommend that the first line of defense is partial resistant. You can look on the seed catalog. There are ratings for brown stem rot. And then some of the seed treatments are pretty good as well for the soil-borne pathogen. So some of the seed treatments that in that fact sheet, um, will be labeled good or very good for brown stem rot. Those are the ones that I probably choose for my operation. So we have that as well.
Okay, thank you much. Aracha, that's all the questions. We do thank you for your presentation. If anyone does have any other questions, Aracha will be on probably a little bit and could answer those if you want to put those in
Yeah,
the Q&A.
I'll stick around. And again, this final slide is just a thank you to our support from funding agency, and again, a big thank you to my lab members and all of the great um, farm directors of the different research stations. So farm managers. So Joe, Lynn, Matthew, Frank and Bob and Matt. So excellent people to work with.